Welcome. Continuing with the biotechnology series, I, Dr. Charu Dogra Rawat, Associate Professor at Ramjas College, University of Delhi, discuss advanced tools and techniques. In it, we will be discussing about the blotting techniques. We had taken up southern blot hybridizations and northern blot hybridization in our last session. Today, we continue with the third blotting technique, which is the western blot analysis. So, the learning objectives for today's session would be to understand the principle and procedure of western blotting, and if the time permits, we will try to understand the principle and procedure of simple polymerase chain reaction, else we will take it up in the next session. The references used only for educational purposes in this series is T.A. Brown, Gene Cloning and DNA Analysis and Introduction, and Advanced Molecular Biology, a concise reference by R.M. Twyman, apart from some of the web series links which are mentioned on the slides. We talked about this blotting compass in our last session when we discussed about the southern and northern blotting, and as we can see, the southern blotting was basically concerned with the DNA, northern blotting is for the RNA, and for protein detection, identification, as well as using it for other manipulative purposes, the western blotting is being done. There are variations to it when we deal with either the template is one of them and the probe or the identifier is something else, then we get different kinds of varieties which are in between. For the eastern one, we generally include the lipids, and as we said, there are post-translational modifications also that are detected. So these are basically all just the variations of it. Primarily, these three techniques are used in biotechnology, which is the southern blotting for DNA, northern blotting for RNA, and western blotting for proteins. In fact, when there is a protein-DNA interaction or there is a DNA-protein interaction, for example, in electrophoretic mobility shift assay, so the similar techniques, range out from all these different blotting techniques. So as I said, today we will be talking about western blotting, which is used to detect specific protein molecules from among a mixture of proteins. This mixture can include all of the proteins associated with a particular tissue or cell type. Now, just as we talked about southern blot hybridization, where we were looking for the region of interest from different DNA fragments. So, there was a pool of DNA fragments and we were trying to identify, locate or isolate, just basically identify first and then isolate a particular DNA fragment which was our gene of interest. In northern blotting also, we take the total RNA from a cell or a tissue type or under certain conditions. As I mentioned that northern blotting is used for basically assessing the expression pattern of a particular gene. And then we tried to identify whether the RNA of a particular gene of our interest was present or not. So we tried to identify that. In western blot, the biomolecule is the protein. So there is a mixture of proteins that we isolate from a particular tissue or cell type, and then we try to detect the specific protein molecules from that mixture. It can also be used to evaluate the size of a protein of interest and to measure the amount of protein expression. So this detection, because I was connecting it to the recombinant DNA technology that we had done in earlier sessions of the series. So to connect that, the detection of the protein is done by western blotting, but other applications also include to evaluate the size of the protein of insert or to measure the amount of protein expression. Now, how do we get this protein sample and how do we prepare it? So, the preparation and the isolation. So, as you can see on the left hand side, there are different types of tissues from where the protein samples can be isolated. The total protein can be from a bacteria, it can be from a type of a cell. When we are comparing different conditions, that a cell undergoes, maybe it is a diseased or an undiseased condition or under certain parameters. So we take those cells and then we isolate the protein from them. Now on the right hand side pattern, it tells us about the lysis, the different types of lysis systems that are used to isolate the total protein samples. So depending upon the type of cell, now different lysis mechanisms are used. We have done the isolation of DNA and isolation of RNA. We covered the isolation of genomic, plasmid DNA, etc. in another session. So for the protein, we do not need to go too much in detail. but we need to talk about these different methods. So there can be a chemical lysis method which is there, where we use different detergents to lyse the cell and entire protein comes out. When we use tissues and cells, we can also use simple mortar and pestle. So we take the tissue, we isolate the tissue, place it in the lysis buffer containing the detergent, and then use a mortar or a pestle to just grind it so that the entire protein lysate comes out, and then we can centrifuge and remove the cell debris. So different kinds of methods are used. Equipments like sonicators are used. So sonicator again, agitate it. So there can be water shaking also, or there can be a specified and dedicated sonicator, for example, containing a probe which is introduced into the cell or tissue containing, with the lysis buffer, and they are sonicated at particular conditions. So with this high frequency sound, the cells burst open and the proteins are released. So the cell lysate can be prepared by n number of mechanisms, and some of the mechanisms are written here for particular cell types that are used. So that is the first step, that we isolate the total protein, and for isolating the protein, you can use different samples, and you can use different types of lysis systems by which you can release the proteins and then collect the lysate to manipulate further. We also add protease inhibitors in the lysis buffer. So apart from the detergents or certain chemicals that cause the disruption of the cells or the tissues, protease inhibitors are very important. So a series of protein inhibitors are given in this table. And these protease inhibitors, so protease is an enzyme that denatures denatures the protein. So we do not want the proteins to be denatured. We want the proteins to be isolated. We do not want it to be degraded. So we use certain inhibitors which are there. So these inhibitors are used at different concentrations, and at different concentrations they tend to inhibit any kind of protease which are present. So as you can see, different types of proteases are also mentioned here with their where it works or on what proteases it works and what are the concentrations that it works as, and therefore it inhibits those proteases and causes the isolation of the total protein. So this is a very, very important component in the protein isolating lysis buffer or the sample which is used. After isolation of the total protein, an important aspect is to quantify the protein, that is to know the amount of protein. Even when we do southern blot hybridization or northern blot hybridization, it is very important to know the initial quantity of the biomolecule we are working with. Because we cannot, I always repeat this, when you use DNA, when you use any biomolecule for manipulation, genetic manipulations, recombinant DNA technology or any other technique of biotechnology, you first need to quantify that biomolecule. Because we do work with volumes, but we design everything using the amount. So because if it is dissolved in water, we will always, water or whatever buffer it is dissolved in, we measure in volumes that how much volume we need to take. But the design of the experiment, the requirement of the enzyme which is going to manipulate this, or what are the conditions, we need to know the amount. So in this also the protein quantification is very important, and two methods are used for, there are many methods, but these are the two basic methods which are used for protein quantification to ascertain the amount of protein which is present in the lysate. So this amount is very important, as I said. For example, if you want to compare two cells, one in which is in a normal condition and one which is in a diseased condition, and you want to detect the presence of a particular protein, or even if you want to detect the presence of a particular RNA. So if you isolate the total RNA, then you must load on the gel equal amount of RNA. Then only you will say that it is either expressed or it is not expressed in the diseased condition. Similarly for the protein, if you are talking about the protein expression or where the protein is formed, then For example, in these two conditions, where there is a normal cell and where there is a diseased cell, or where there is an experimental set that you have cell that you have put under certain conditions, whether that protein is detected in that sample, or if it was detected in the normal cell and it is not detected anymore in the modified cell or experimental cell, then for that you need to load the same amount on the gel, the same amount of protein on the gel. Then only you can compare that, okay, it was present in the normal but it is not present in the experimental condition, and vice versa. So this protein quantification becomes very important. So two methods. One is a very common method which is called as the Lowry method. So it talks about the reduction of the copper ions, and therefore it gives a particular colored substrate when this change occurs, and that colored substrate can be identified at 750 nanometers. So you can use a simple colorimeter to basically identify the presence of a protein. So it is quite an inexpensive and a routine technique that is used, but it can detect only up to 0.01 to 1 mg per ml. So the detection limit is not very low, but it is an advantageous method. The disadvantage is basically that because of the buffer systems and different systems, the protein can be denatured, or you cannot, as I said, there is a limitation, you cannot detect smaller proteins, etc. So that comes in between. Then there is a Bradford assay. So in Bradford assay, there is a Comasi gel stain 250 which is used to stain the protein. And this basically, as when association with protein causes the dye to shift from the red to a blue color, and this can be again detected. So depending upon the amount of protein that is present, whether it is present or not, we can detect this system. Again, it is a very good system that is very routinely used. So we get kits, basically we get these solutions, we add it into the lysate and we measure the wavelength in the spectrophotometer or in the colorimeter, and therefore we can quantitate the amount. So for both the things there has to be a standard curve, and through that standard curve, so now it is feeded in the softwares also. So in that standard curve, the amount of the unknown, which is our protein, we can extrapolate and get it. So this quantification becomes very important because the starting amount need to be known. Although we will use and measure in volumes, but we design in amount. Going ahead with the Western blotting procedure, the first aspect of a Western blot is to run a gel. The same thing we did with DNA and RNA. I just keep talking about DNA and RNA just to create a parallel of these techniques. These are blotting and hybridization techniques. So in the blotting and hybridization techniques, the similarity is this. So there also you isolate the DNA, you quantitate the DNA, and then you run it on an agarose gel. Similarly for RNA, you isolate the RNA, you quantitate the RNA, and you run it on an agarose gel. And from that gel, it was transferred onto the membrane. So principally, the blotting technique is the same. So here also, you have isolated the proteins, you have quantitated the proteins, now you run it on a gel. But you do not run it on a simple agarose gel, but we run it on what is called as a polyacrylamide gel electrophoresis. And this polyacrylamide gel electrophoresis, we also use sodium dodecyl sulfate. So it is termed as STS-PAGE. There are other electrophoresis systems also, which is like native PAGE, etc. But we are not going into detail with that because we are doing a general concept of the Western blot, and that we might take in our subsequent sessions. So we are talking about the simple procedure, that how do you do it in a lab, or what, how do you need to design it. So instead of the agarose gel, which has got a higher pore size because of the concentration, whatever you do, you get a little large, so you cannot isolate smaller fragments. You do a polyacrylamide gel electrophoresis system. So the principle of this polyacrylamide, the SDS polyacrylamide gel electrophoresis, is that when you have isolated the proteins, now you know the structure of proteins, they are folded together. They are the different polypeptide chains can be bound by the disulfide linkages. So that is basically the tertiary structure of a protein molecule. So whatever proteins you have been isolated, they will be folded, and in that folded manner there will be disulfide bridges which would be there, and there will be a pattern of this thing. So what we do is we prepare this sample. We just do not, the isolated sample, we just do not load it on the gel directly. We prepare this by basically adding chemicals such as beta-mercaptoethanol. So this beta-mercaptoethanol basically breaks these disulfide bridges, they linearize it, they unfold it. So sodium dodecyl sulfate is causing the unfolding of the protein into polypeptide chains, and then it also masks these chains. So as you know, the proteins are basically amphoteric compounds. They can have positive charge because a protein is a chain of amino acids. Amino acids can be basic, can be acidic. It can have a positive charge, it can have a negative charge. So as a whole, the protein may carry some positive charges, some negative charges. So basically this SDS, which is a negative charge, masks the entire polypeptide chain, so that when we try to fractionate these polypeptide chains, the fractionation is only based on the size, because now it has a homogeneous charge. Otherwise the charges, the net charge on a protein molecule can differ. One molecule can have a net positive charge depending upon how many amino acids containing positive charge is present, and similarly for the other protein. So they can be negatively charged and they can be neutral also. So we add SDS. So what does SDS do? It unfolds the protein. Beta mercaptathol basically removes the disulfide bridges. So you get polypeptide chains of different sizes and all covered by this negative charge. So the fractionation or the differentiation when we do will only be based upon the size and not the charge. So in native gel electrophoresis it is different, where the separation occurs because of the charge is to mass ratio, but in this we generally do the separation only based on the size or the mass. So this is the basic principle of the sample preparation for SDS page that we do. Then next what we do is, this is the setup or the apparatus. So various companies give these kind of setups. So as you can see, this is a casting trays. So because we run a vertical gel, now polyacrylamide we make very thin gels. All this is basically for the resolution. The vertical resolution is much more than basically the horizontal resolution, because in vertical the gravity also plays a role. So when there is an electrode, so you can set the gel in these kind of a casting trays which are there. So there are two glass plates, and in between these two glass plates, so the thickness is really small in mm. The gels are really small. So you can pour the solutions and you can let it set. Once it is set, then it can be run in these kind of buffer tank systems. These tanks will contain the running buffer. You can add the gel in here and then you can load the samples vertically. So these are the combs, these are the glass plates, and then you can just run your samples. Obviously you will run a marker along with it if you want to know the size of the different protein molecules, and you can just run this gel. You can connect it to a power pack supplying the power, and you can run the gel. So again this will be electrodes, and there will be different electrodes, and because it is negatively charged, all the peptides are negatively charged, it will run towards the positively charged molecule. So these setup provided by various companies, but these are the basic components of a setup for a gel running system. Now the polyacrylamide gel, it is prepared by mixing the acrylamide with bisacrylamide to form a cross-linked polymer network when the polymerizing agent, which is the ammonium persulfate APS, is added. Another component, which is the timid, catalyzes the polymerization reaction by promoting the production of the radical by APS. At this stage it becomes polyacrylamide. So what we do is that, for example, the DNA gels, the agarose gels were made up of agarose. So agarose for different concentrations were mixed in the buffer, which is the TBE buffer or TAE buffer, which we used to use, the tris borate EDTA or tris acetate EDTA buffers. We add whatever concentration we want to make agarose into it, and we heat it. So when we heat it, it gets mixed, and then we set it at room temperature, so it sets, the gel sets, it becomes gel-like structure. For acrylamide, polyacrylamide gels, you use two components, which is an acrylamide and bisacrylamide. Now their concentration and the concentration of this polyacrylamide will define the pore size, just like in agarose gel. So for example, if we use a higher concentration of agarose, the pore size will be smaller. So the similar is in this case. So higher the concentration of acrylamide we'll use, higher will be the resolution and lower will be the pore size. So depending upon that, we require APS for polymerization, and immediately when we need to set, we add just the timid, and within a minute, even less than a minute, the gel will set, and we leave it for some time on that casting unit where we were trying to set the gel. So this is basically a polyacrylamide gel. This is the buffer system it shows, and this is a vertical gel electrophoresis I said, and it utilizes a discontinuous buffer system. So discontinuous buffer system is that you can observe that there are two different kind of gels which are used here. One is called as a stacking gel and another is called as a resolving gel. So this vertical gel has two different gels where the concentration of bisacrylamide and acrylamide are different and the pH is a little different. So this is why, because when you add the proteins, they all stack together. This is a stacking gel. It has a larger pore size, and because of a particular pH, all the proteins, they stack together tightly in a bond. And when they enter a resolving gel, there is a change in the pH as well as there is a change in the pore size, and therefore they get resolved and separated inside the resolving gel. So the two different kind of gels are used, and there is a discontinuous buffer system. So discontinuous buffer system means that the buffer which is used to make the gel, the ions that is contained in the buffer is different from the running buffer. If you can recall, in agarose gel electrophoresis, we have a special lecture on that, visualization of genomic and plasmid DNA by agarose gel electrophoresis. You can go through that. The gel buffer which was used to make the agarose gel was TBE or TAE, and the same buffer was used to run the gel. So that is a continuous system. But in here we use a discontinuous buffer system. We'll talk about that, what happens when we use discontinuous buffer system subsequently. Now, to obtain the optimal resolution of proteins, a stacking gel is cast over the top of the resolving gel. The stacking gel has a lower concentration of acrylamide, for example 7% for large pore size, a lower pH, for example around 6.8, and a different ionic content. So the resolving gel is around 12%, it has got a smaller pore size, a little higher pH which is 8.8, and a different ionic content. This allows the proteins in a labeled sample to be loaded sample to be concentrated into one tight band during the first few minutes of electrophoresis before entering the resolving portion of the gel. So what happens is that in polyacrylamide gel electrophoresis, we utilize two different types of gels having different ionic content, different acrylamide concentration and different pH, so that the total proteins that we have isolated, they first stack together very tightly in a band, and as they enter the resolving gel, they start separating. So this kind of a system gives the better resolution or an optimal resolution of proteins which is required, because we get very different size of the fragments when we have unfolded the protein and created the polypeptides. So we continue with the discontinuous buffer system in our next session. Thank you.